Milk is a source of nutrition for the infant. It is a complete source of nutrition. The infant needs no other food for months or years of its life. But milk is more than that. Milk contains mRNAs and microRNAs. In fact, milk has the greatest concentration of mRNAs and microRNAs of any body fluid. The microRNAs are released in fat globules and exosomes, small vesicles. These exosomes primarily being produced by myoepithelial cells. The purpose of miRNAs is not to serve as a source of nutrition, but rather to regulate gene expression in the cells of the infant. It had been assumed that anything produced in milk would be digested in the digestive system of the infant, in the stomach and the small intestines. But exosomes resist digestion. They are absorbed into the cells of the infant through endocytosis, and then can even be found in the blood of the infant circulating throughout the body, where the miRNAs can then be taken up by diverse cells throughout the body, where they can regulate gene activity. In this example, the infant's cell has transcribed the gene P53, a very important gene regulating cell division. But the microRNA 125B from milk then binds to it, which will inhibit its translation, making less protein likely to produce, or even cause the degradation of this transcript. Human milk includes miRNA 148, which will bind to the gene transcript of the DNA methyltransferase 1 gene in infant cells, inhibiting it. There are other microRNAs in human milk, many of which influence cells of the immune system and change their activity and gene expression. This raises a whole host of questions. So if one child is breastfed and another child is on formula which lacks these miRNAs, are there differences in gene expression between these two infants? Might these be responsible for the overall differences in the risk of allergy, asthma, diarrhea, obesity, and other conditions observed between breastfed infants and infants on formula? Not only does human milk contain microRNAs, so does the milk of cows. They are not identical in quantity and in the types of miRNAs, but some are identical in sequence to human miRNAs. This raises a question. Since microRNAs survive pasteurization, but not the fermentation to produce cheese, are adults and children who drink milk then receiving instructions on gene expression made by a mother cow intended for an infant cow? Are some of these instructions inappropriate? Thus, the turning off of P53, which is sometimes referred to as the guardian of the genome, preventing cancer by its inhibition of cell division, is the effect of milk on the expression of this gene negative for adults? The DNA methyltransferase gene affects the methylation and epigenetic control of many genes. Is drinking milk maladaptive for the expression of this gene in adults?